Yeah. So this is first tutorial session, and so my name is Kundan Solanki. Okay, I am a PhD student at IIT Indore, and I am one of your TA for this course. And basically, the idea for this tutorial session is to discuss the doubts, okay, regarding the regarding the content which you have accessed throughout the week, okay, through this week, the lectures which you have gone through. So if you have any doubt, then you can ask in this tutorial session. Okay, so we can discuss regarding that. And another thing regarding this tutorial session is to solve the assignment question. Okay, so you have already submitted the assignment. The due date was it has already extended the due date. Okay, so you had submitted the assignment question for the week one, and in this session, basically, what we'll be doing. So we'll be looking at that assignment question and we'll be try to solve it. Okay. So it is rather a simple question, but we will look it one by one and the most probable answer and the other options also we will see. Okay. The why other options are not an correct option. Okay. So we'll be discussing that and apart from that we'll be discussing first we'll be discussing about the course content which you have gone through in the first week. Okay. So we'll be looking at that thing first. We will quickly revising that thing of the content of week one, and then we'll be moving to the assignment questions. Okay. So I will share my slide. Okay. So in summary, if I just quickly revise, then uh, first you had seen. Is my slide visible to all of you? Can anyone reply?
Yes, sir.
Okay. Okay. Thank you. So in the first uh, in the first week, we basically what we studied was regarding the basic idea of the cell biology. Okay, so when we talk about cell biology, so we talk about the cell. Okay, and in the cell there are various components associated. Okay, in the cell when we talk about then there is a first of all, at the outer side, there is a plasma membrane, okay, which is a membrane which protects the cell from harmful other outside effects, okay, outside environment, as well as it mediates for certain physiological functions to the cell, okay, by the penetrations of ion and other solute molecules, okay, solute and solvent molecules. So it acts as a semi-permeable membrane. So outer side it has a plasma membrane. At the inner side, there are various other organelles present inside the cell. Okay, such different organelles depending on the organism. So if you talk about eukaryotic organism, then there are various organs such as mitochondria. Okay, nucleus. We have nucleus, mitochondria, ribosomes, and all the other things. Okay, which helps in the functioning of the cell. So that includes the structure of the cell. Okay, and when we talk about the function of the cell, when we discuss about the function, then there are various functions associated inside a single cell. Okay, so in a single cell there are many other process, metabolism process. When we talk about metabolism, then we discuss about the glycolysis pathway and the Krebs cycles, okay, which converts the glucose molecule into pyruvate, then pyruvate gets converted into acetyl CoA, okay, and which goes through the Krebs cycle and generates energy, okay, in the form of ATP and other reducing equivalents such as NADPH and FADH2, okay. So this constitutes the function of the cell in the mitochondria as well, which we already know it is a powerhouse of the cell. Okay, so it generates the ATP molecule. So various metabolic functions occurs in the single cell, and apart from the function, there is a reproduction of the cell as well. Okay, so each and every cell has an ability to replicate itself. Okay, to undergo the cell cycle process and replicate itself into its different daughter cells. Okay, so the reproduction capacity is also there for a single cell, and apart from that, there is a communication. Okay, intra and intercellular signaling between the cell as well, and as well as inside a single cell, various cell cycle factors. Okay, which interacts with each other, various proteins which interact with each other to promote the various physiological process inside the cell. That can be cell division, or for example, transcription and translation of certain proteins or genes which are necessary for the maintenance of the cell, okay, to maintain the homeostasis. So when we talk about cell biology, we discuss all the aspect of the cell, such as structure, function of the cell, as well as reproduction, how it is reproducing and undergoing cell cycle, as well as communication, intracellular signaling occurring. Okay. So if I talk in basics, if I talk in preliminary, if I talk about the cell theory, so initially, what we currently know is that cell is the fundamental structural and functional unit in the living organisms. Okay, so whether it can be unicellular, multicellular. Okay, so certain unicellular organisms contain a single cell in which it helps in the various function, and an organism is at least made up of one cell, and for multicellular organism there are more than one cell. Okay, and cell arises from the pre-existing cell by cell division, and it carries the hereditary information that passes on to the daughter cell during the cell division. Okay, we know the genetic material, that is DNA. DNA is the genetic material inside the cell, and during the cell cycle division, that genetic material, each copy is passed to the daughter cell, and that is how the hereditary information is transferred from one cell to another. Okay, so apart from that, each and every cell has a similar chemical composition and structural organization. So in a single organism, when we talk about the eukaryotes, okay, so in eukaryotes as well, when we segregate, as we will see in the later slide, so there are photosynthetic or eukaryotes and there is non-photosynthetic eukaryotes. Okay, so in both the kind of organisms, the composition of the cells is similar. Okay, as well as structural organization. It will be having the same number of nucleus, ribosomes, mitochondria, and all those things. So the chemical composition and structural organization doesn't change in the organism. Okay, so it remains the same. And there is a flow of energy within the cell. So when I talk about the flow of energy, then we talk about the ATP molecule. So generation of ATP during the mitochondria. Sorry, is there in doubt? Yeah. So energy flow within the cell, that is generation of ATP and the utilization of this ATP in various metabolic process. Okay, so this is regarding the cell theory. So when we discuss about the prokaryotes and the eukaryotic organism, so yeah, is there any doubt, anyone? Okay, so if you have any confusion in any of this slide, you can unmute yourself and ask in the between as well. Okay, it's completely okay. Okay, so you can unmute and ask at any time point. So when we talk about the prokaryotes or its eukaryote organism, so in prokaryotes, generally when we talk about prokaryotes, it is a unicellular organism and it ranges in the size. It is a very small in size. Okay, point 2 micron to 2 micron in diameter. The cell size. Okay, the cell size is point 0.2 micron to 2 micron in diameter, and it doesn't contain the nucleoid. Okay, and it doesn't have the nuclear membrane. So although nucleus will be there, but there will be no nuclear membrane, and there will be no nucleoid present. Okay, so nucleoid, as we'll be seeing, so nucleoid is an organelle which constitute the ribosomal synthesis and assembly of ribosomal RNA. Okay, so ribosomal RNA are produced and assembled in the nucleoid, but in the prokaryotes, the nucleoid is not present. Okay, and apart from that, in the prokaryote, DNA is circular. Okay, and the cytoplasm has cytoplasm has a ribosomes. Okay, ribosome is contained in the cytoplasm, and mitochondria and centrosome is absent. So this constitutes the prokaryotes. In prokaryotes, generally bacteria and archaebacteria. Okay, so bacteria such as E. coli and all the other bacteria. Okay, so that and as well as the archaebacteria, which is genus belonging to archaea, uh, are also included in the prokaryotes. Okay, and in the eukaryotes, it is generally multicellular. Unicellular eukaryotes are also present, such as yeast. Okay, so yeast is a unicellular multi eukaryote, and it ranges in the size from 10 micron to 100 micron in diameter. And there is a presence of nucleus, and it is a membrane bound. Okay, so nucleus is a membrane bone compartment which contains the DNA in its or DNA inside it. Okay, and the DNA is linear compared to the circular DNA in the prokaryotes, and the cytoplasm contains ribosome and other organelles. Okay, such as endoplasmic reticulum, lysosome, Golgi body, and peroxisome, and all the other functions or the other organelles are present. Okay, which has a which individually has a very distinct function inside the cell. Okay, and mitochondria and centrosome are also present inside the cell. So centrosome basically helps in the replication during the cell division process. Okay, during alignment of the mitotic spindle. Okay, so centrosome plays a role in that part. So this is regarding the eukaryotes, and in eukaryotes when we talk, there are examples like animal, plants, yeast, and protist. Okay, so then we will yeah. So we talk about the various bacteria. Okay, the bacteria which we use currently in the model organism we use in the bacteria is E. coli. Okay, so E. coli we use as a model organism, and because it is relatively simple to culture. Okay, and many other things are known. Okay, the gene organization and the function of this E. coli and all the other things are already well established. That is why it is used as a model organisms in the molecular biology and cell biology. Okay, to study its function. So in the E. coli, in general, when we talk about bacteria, there are two kinds of bacteria, as you already know. Okay, so one is gram-negative and another is gram-positive bacteria. So there is a certain cell. Basically, the difference lies in the constitution of the cell membrane. Okay, so in gram-negative bacteria, basically, the peptidoglycan layer is very thin. Okay, so here you can see in the violet portion. Okay, so there is a thin peptidoglycan layer, and the outer membrane, there is a presence of the outer membrane. So beneath the outer membrane there is a peptidoglycan layer. Okay, stacked between the outer membrane. So as a result, when we use the crystal violet staining, okay, so crystal violet staining is a which we discuss in the gram staining. Okay, gram, to distinguish the gram negative and positive bacteria, we basically do the gram staining. Okay, so in gram staining basically we use the two dye, basically crystal violet and safranin. Okay, so in crystal violet, what it will do, it will stain the peptidoglycan layer of the bacteria. Okay, so if the bacteria is having the high peptidoglycan layer, it will stain that layer. Okay, so in gram-positive bacteria, basically the peptidoglycan layer is very thick and it doesn't have any outer membrane. Okay, outside the bacteria membrane. So as a result, the crystal violet, by using the crystal violet stain, the peptidoglycan layer will be getting stained, and after that, after washing, we use a safranin dye. Okay, so if the peptidoglycan layer is not stained properly, as in case in the gram negative bacteria, the safranin will stain the outer membrane. Okay, of the gram negative bacteria. So that is how we distinguish between the gram positive and negative. In positive, basically we observe is the violet staining, and in the negative we observe is the pink staining due to safranin. Okay, so that is how we distinguish between the gram positive and negative bacteria. And apart from this, there is a photosynthetic bacteria as well, which we call as a cyanobacteria. Okay, so cyanobacteria basically derives the energy from using the sunlight as an energy source. Okay, so it has a photosynthetic pigment present. Okay, inside the cell, which converts the cellular energy from the sun into its energetic component. Okay, to derive the ATP formation. Okay, and formation of glucose molecule basically. It is unstable. Yeah. So when we talk about the molecules, after that, okay. So in the molecule part, we discuss about the various elements present inside the cell. Okay. So elements in the human body generally constitutes of carbon, hydrogen, nitrogen, and oxygen. Okay. So these four elements makes up the 99% of the elements in the human body. Okay. So uh, out of carbon, hydrogen, nitrogen, and oxygen, macromolecules are constituted, such as proteins, nucleic acids, and lipids, and all those things. Okay, which consider the majority part of the cellular compartment. Okay, cellular organization, and these atoms basically helps in the covalent bond formation, and that is why they are helpful in generation of the macromolecules. Okay. And apart from that, other elements and other trace elements are also present in minor quantities, such as sodium, potassium, magnesium, calcium, phosphorus, sulfur, chlorine, and other trace elements such as uh, iodine, vanadium, chromium, molybdenum, manganese, cobalt, and all those things. Okay, so these are all the trace elements which are present and which are required as a because certain enzymes requires this element as a cofactor. Okay, for example, if we talk about hemoglobin, so hemoglobin requires iron as a cofactor, okay, without which it will not function properly. Okay, so apart from that, sodium, magnesium, and also potassium are also required for different enzymes. So these cofactors are required, and that is why its importance is it is essential to have these elements. Okay, and organic molecules when we talk, then the small molecules and macromolecules. Okay, the sugar is a small molecule, and if we constitute sugar molecules, okay, if there is a repeating units of sugar molecule, then it constitutes the polysaccharides. Okay, large polysaccharides. And similarly with the fatty acids, if we constitute a single fatty acid, okay, when there are more than one single fatty acids, then it constitutes the lipids and membranes. So lipids and membranes constitute the fatty acid. Similarly, in the proteins are made up of number of amino acids, okay? And in the nucleic acids, there are presence of nucleotide. So, so these are the unit of a macromolecule, okay? So for amino acid, it is protein. For nucleotide, is nucleic acid, okay? And certain inorganic ions, for example, uh, potassium and calcium and all those things are essential for its maintenance. And apart from that, also the bonds which are present inside the between these atoms. Okay, so certain ionic bonds, hydrogen bonds, van der Waals attraction. Okay, and hydrophobic interaction between the molecules are important in maintaining the structure of that molecule. Okay. So, for example, if we talk about the hydrogen bond, which is an important bond, okay, forming between the elements, so in the water molecule itself, if there is no hydrogen bond between the water molecule, then it will not maintain the structure of that water molecule. Okay, and it will not maintain its properties. When we talk about the water molecule, that it has a specific properties. Okay, it has certain boiling point, it has certain melting, it has certain specific heat of capacity and latent heat of vaporization and all those things. Okay, the entropy and all those associated thermodynamic parameter. So that thermodynamic parameter is basically constituted by formation of various type of bond in water. In case of water, when we talk, it is constituted by hydrogen bond. And if there is if this property is not there in the water molecule, then it will lose its property. Okay, individual property. Just a minute. There is some just a minute. I don't know why it is happening like that. Okay. Yeah, so after the elements, we talk about the small organic molecules. Okay, so organic molecules basically, when we talk about organic molecules, so it contains the carbon atom. Okay, carbon, hydrogen, and oxygen. Okay, so if the molecule contains a carbon atom, it is an organic compound. Okay, yeah, so organic molecule constitute the major part of the cell, as we discussed. Okay, so proteins and protein contains amino acids. Okay, so in amino acids, it are made up of carbon units, and there are also nitrogen and hydrogen are also present. Okay, so these constitute amino acids. This constitute the single amino acids. Okay, with the carbon and hydrogen atoms. And when you talk about carbon atoms, so carbon constitutes the major part of this macromolecule. Okay, and in the carbon atom as well, when we talk about carbon, then it has an sp2 co-conform hybridization capacity. Okay, so in the outer shell electron, there are in the in its outer orbits there are four electron. Okay, four unpaired electrons. Okay, it has an sp2 or sp2 hybridization state. So it can form because of there is four valence electron in the outer shell of the carbon atom, it can make four hydrogen bonds. Okay, four hydrogen bonds with the surrounding hydrogen atom. Okay, so carbon makes four covalent bonds between the carbon or the hydrogen atom. Okay, and this type of bond formed between the carbon carbon are very strong. Okay, very strong covalent bond, and due to this covalent bond formation of the between the carbon carbon atom, the rigidity of the macromolecule is conserved. Okay, there is a high rigidity in the amino acids and all the other macromolecules by formation of this covalent carbon carbon bond. Okay, which requires high enthalpy. Okay, to break this hydrogen bond, break this covalent bond. Okay. And so yeah, so if I talk about the amino acid, which is a building block for the macromolecule, so in the amino acid as well, the carbon atom is there, and in the side chain, which differs in the side chain. Okay, individual amino acid differs with the side chain. So in the side chain, what are the amino acid present? So if there is a hydrogen present, then that is a simple amino acid. Okay, so it is a glycine. When we talk about glycine, though, it is a simple amino acid. So it doesn't contain any side chain. But when you talk about tyrosine, so it contains an aromatic side chain. Okay. Uh, when we talk about the acidic amino acid, okay, for example, over here there is an amino acid group. So when you talk about acidic amino acid, it contains NH3, okay, in the side chain, which promotes the acidic nature of this glutamine and aspartate, for example, okay, which is acidic amino acid, which contains an NH3 group in the side chain. So as well as here you can see various non-polar, polar, acidic and basic amino acids. Okay, so depending on the side chain, the polarity of the amino acids are formed. Okay, so in the non-polar amino acid, basically it contains the aromatic side chain which are non-polar, as well as certain entity carbon compounds. Okay, besides amine or the carboxylic group. Okay, the non-polar amino acid we talk about alanine, valine, leucine, isoleucine, proline, phenylalanine, methionine, tryptophan, glycine, and cysteine. Okay, so these are non-polar amino acid. And in the polar uncharged amino acid, there is an asparagine, glutamine, serine, threonine, and tyrosine. Okay, so this polar means polar uncharged means it contains a side chain which contains the uh, NH3. Okay, NH3 or COO minus. Okay, carboxylic or amine group in the side chain. And when we talk about acidic and basic, so that includes like aspartate and glutamate, and in basic, lysine, arginine, and histidine are present. Okay, so these are the classification of the amino acids. And yeah, so next, when we talk about the macromolecules, so in macromolecules, we have polysaccharides, nucleic acids, and proteins. So polysaccharides are made up of are made up of are a type of carbohydrates. Okay, so it is made up of it is made up it is made up of it constitutes the carbohydrate moiety of the. Okay, so in polysaccharide, when we talk about the example is like glucose, okay, fructose, and all those things which are made up of which has an saccharide group. Okay, sugar molecules present inside that, and nucleic acids are made up of nucleotides, and proteins are made up of amino acids. So when we talk about the protein, so there are four kinds of protein: primary, secondary, tertiary, and quaternary structure. Okay, so in primary structure, when we talk about the primary structure, so each amino acids are linked by the peptide bond. Okay, so peptide bond are formed between the amine group of the amine group of the one amino acid and the carboxylic group of the adjacent amino acid. Okay, so due to the condensation reaction, the water molecule is lost and then peptide bond is formed between the two amino acids. Okay, and it occurs in the single chain. And after a sufficient peptide chain is formed, it forms a structure depending on the conformation and type of amino acids. Okay, so it can form alpha helices or beta pleated sheets. Okay, so both of these structure can be formed in a single polypeptide chain. Okay, depending on the amino acid configuration. Okay, so these are the called the secondary structure. And in the tertiary structure, basically we talk about the single domain of a protein. Okay, so one domain which contains a beta sheet as well as alpha helices constitutes the tertiary structure. Okay, so in the tertiary structure, when we talk, so in polypeptide chain, generally the bond formation is a peptide bond. Okay, there is no presence of hydrogen bond or uh, hydrophobic interaction and all those things. In alpha helices, we observe the hydrogen bond formation as well as hydrophobic interaction. There is not much hydrophobic interaction, but hydrogen bonds are most prominent in the secondary structure. When we talk about the tertiary structure, there are more non-covalent interaction, okay, such as hydrophobic interaction as well as other ionic and van der Waals interaction. And in the quaternary structure, basically it is made up of multiple domains of a single protein. Okay, for example, in the case of hemoglobin. Okay, so hemoglobin is hemoglobin constitutes the multiple domains inside a single RBC molecule. Okay, so it has a multiple domain. So a single domain is called a tertiary structure, but if it considers a multiple domain and it is linked with the single cofactor, okay. In the case of hemoglobin, it is linked with iron. So multiple domains are linked with the iron molecule, and it constitutes the quaternary structure of the protein, okay. And each and every domain function dependent on each other, okay. So that makes up the quaternary structure of the protein. So these are the four structures kinds of protein. Okay, depending on the domains which are associated. So next, when we talk about the plasma membrane components, as I talked earlier, that in the cell, the plasma membrane plays a very important role. Okay, okay. So when we talk about the plasma membrane, the plasma membrane is designed such that it will not be able to, it will not allow the penetration of any of the ions. Okay, so compounds such as ions and other charge molecules. Okay, the charge molecules and the ions will not penetrate inside the plasma membrane. Okay, and but certain compounds such as hydrophobic compounds. Okay, hydrophobic compounds can easily penetrate through the cell membrane because the cell membrane is hydrophobic in nature. And certain hydrophilic compound which have a polar charge molecule. Okay, such as polar charge molecule and ions, they are hydrophobic in nature. So that they will not allow the passage of that compounds. So plasma membrane are also made up of lipids basically. Okay, so that lipids are called phospholipids. So when we talk about the lipids as well, in the membrane lipids, there are three kinds of lipids. Okay, so one is phospholipid, another is glycolipids, and third is cholesterol. Okay, so when we talk about the phospholipid, so in phospholipid generally is made up of two hydrophobic fatty acid chains. So here you can see. Okay, just a minute. Okay, so here you can see this is the fatty acid chain, and it is linked with the glycerol group. Okay, so glycerol group is attached in the glycerol. So in the glycerol there are three carbon atoms. Okay, arranged. So if I show it over here, so this is one, two, and three. Okay, so at this first and second carbon, the fatty acid side chain will be attached. Okay, and in the third carbon, basically, this third carbon will be uh, will be attached to the phosphate group. Okay, so this carbon will be attached to the phosphate group. Okay, so this carbon will be attached to the phosphate group, and this phosphate group again it will be attached to some polar molecules. Okay, such as choline, serine, ethanolamine, or inositol. Okay, so I will simply write in the abbreviation form: choline, serine, ethanolamine, and inositol. Okay, so this whole component constitute basically the phospholipid, a single phospholipid. Okay, so when we talk about phospholipid, this part is common. The glycerol subunit as well as the phosphate group attached part is common. Okay, that is why it is called phospholipid. Okay, because phosphate group is attached over here. That is why it is called phospholipids. So the changes is in the polar uncharged molecule. Okay, polar molecule which is attached with the glycerol moiety. So if it choline is attached, then it is called phosphatidyl choline. Okay, phosphatidyl choline, in which in the RBC basically it contains 17%. Okay, phosphatidyl choline. If the serine is attached in place of choline, it is called phosphatidyl serine. Similarly, phosphatidyl ethanolamine and phosphatidyl inositol. Okay. Okay, so these are the constitute of the membrane lipids in the phospholipid part when we discuss about the phospholipid. And when we talk about the sphingomyelin, so sphingomyelin is also present in certain cases. So in sphingomyelin, basically serine acid residue is present instead of glycerol. Okay, so this is basically the glycerol side chain. Okay, apart from the glycerol side chain, over here serine will be present. Serine or moiety will be present. So that constitutes the sphingomyelin moiety. Okay, so this constitutes the phospholipid. In the glycolipid part, glycolipid is also a membrane component, and glycolipid generally has a two hydrocarbon chain. Okay, so over here we discussed about the fatty acid chain. In in uh, in spa, beside fatty acid chain. Okay, in terms of fatty acid chain, the hydrocarbon chain will be present. Okay, in case of fatty acid chain, in place of fatty acid chain, and that hydrocarbon chain will be linked to the polar group, okay, which contains the carbohydrates. So carbohydrates, carbohydrate moiety chain, okay, carbon atoms will be present. Okay, carbohydrate moiety, and in with the carbohydrate, the two hydrocarbon chain will be linked, and that is the glycolipid part. Okay, the glycolipid constitute only the 3% of the molecule of membrane lipids, and another is cholesterol. So cholesterol has a four hydrocarbon chain. Okay, so as you can see in this structure, so this is the cholesterol molecule complete. Okay, so it has a four hydrocarbon chain, and which are hydrophobic. So this part is hydrophobic, and one end is one in the at the one end there is an OH group present. Okay, and at the other end there is a carbon chain. Okay, carbon chain present over at the other end. Okay, so this whole molecule at this whole part is hydrophobic, but this OH group is hydrophilic. Okay, so when we talk about cholesterol, it is slightly hydrophilic in nature. Okay, slightly hydrophilic, but most of the part is hydrophobic. Okay, so when we talk about the cholesterol, so what role does cholesterol play? Does anyone can anyone give an answer for that? So in the membrane lipids, generally, in the membrane lipid, basically most of the part are there. Okay, such as phospholipids and glycolipids and all are there, but there is also cholesterol present. So do you know what role do cholesterol play in the membrane lipids? Can anyone answer that? You can unmute yourself and answer. Anyone? Any idea, anyone? Yeah, so cholesterol basically helps in the maintaining the rigidity of the plasma membrane. Okay, so at the at the certain junction of the membrane lipids, okay, in the plasma membrane, there is presence of cholesterol. So what cholesterol do is that it maintains the rigidity and flexibility of the plasma membrane. So when I talk about the flexibility, so in case of like when you talk about the there is a polar in the arctic region, okay, in the polar region, basically certain animals which are present over there, okay, which are prone to extreme conditions, okay, which can tolerate up to minus degree temperature, okay, very below cold temperature. So in that case, the cellular component, the plasma membrane can get rigid due to the freezing condition, okay. So due to the freezing condition, the plasma membrane can be very rigid in condition. The cell membrane can be very rigid. But if the cell membrane gets rigid, then it will lose its function. Okay, so at that time, cholesterol maintains the fluidity of the plasma membrane. So it will not allow the cell membrane to become very rigid during the very low temperature. Okay, which is observed in the animals present in the at the polar region. As well as the other thing is at very high temperature. Okay, observed in the certain environments at very high temperature, it will also not allow the disruption of this plasma membrane. Okay, so if you heat the plasma membrane, okay, there is a very high temperature, then there is a chance that plasma membrane lipids present in the plasma membrane can get disrupted. Okay, and it will lead to the dissociation, okay, of the plasma membrane. But cholesterol can maintain the flexibility, rigidity of this plasma membrane during that condition. So it can hold the plasma membrane intact, and it will not promote the disruption of the plasma membrane at a certain temperature. Okay, but after that, if the temperature is too high, by which the cholesterol also cannot handle that flexibility, then it will get disrupted. Okay, so that is the role of cholesterol present in the plasma membrane. Okay, so you need to remember that thing. Okay, and cholesterol generally has a polar self-sealing bilayers. Okay, it is an amphiphilic molecule. So the polar molecule which are present in the lipids. Okay, so polar head, which are the head region, which is made up of choline, serine, ethylamine. So they are polar. So it is present in the outer part of the membrane, and the hydrophobic fatty acid side chain. Fatty acid chains are present inside the inner part of the membrane. Okay. Yeah. So now the distribution. So this is very important. Plasma membrane distribution. So in the distribution part, when we talk, so the plasma membrane is arranged such that it has a certain lipids present at the outside as well as the inner side of the plasma membrane. Okay. So when you talk about the outside of the plasma membrane, there are presence of glycolipids, phosphatidylcholine, sphingomyelin, cholesterol. Okay. And in the inside, there is presence of phosphatidylserine and phosphatidylethanolamine. Okay. So this distribution of these groups, okay, is essential to maintain the functioning of the plasma membrane. Okay, so for example, in the apoptotic condition, if the cell undergoes the apoptotic condition, at that time the phosphatidylserine present inside the cell gets translocated at the outside of the cell membrane. Okay, so if more number of and the phosphatidylcholine and sphingomyelin present outside will be get translocated to the inside. So if the phosphatidylserine is higher in number at the outside of the membrane, that indicates that activates the signaling that the cell is undergoing the apoptotic condition. Okay. So during the apoptosis, this there is a change in the membrane lipid. So that is why the distribution of the plasma membrane is important. Okay, functionally important inside the cells, inside the cell. So there is also mobility associated with the phospholipid. One is like lateral diffusion. So this phospholipid bilayer can move laterally. Okay, as well as the hydrophobic fatty acid chain. can move along its own axis as well, okay, at its own position. And there is also rotation found in a single fatty acid molecule, okay, a single phospholipid molecule. There is also a rotation found. So these three movements are common: rotation, flexion, and lateral diffusion. But the flip-flop movement, as I told, that during the apoptotic condition, this phosphatidyl serine will be translocated to the outside. So this is done by the flip-flop movement, and it is very rare. It occurs only during the apoptotic condition and certain condition during the cell starvation phase. Okay, and during that time only the flip-flop movement will occur. Apart from that, these three movements are common. Okay, so this is regarding the plasma membrane lipids and the plasma membrane organization. Okay, so now when we talk about the plasma membrane, then we talk about the movement of certain molecules outside and outside to the out from the outside to the inside, as well as from inside to outside. Okay, so movement of molecule to and fro from the plasma membrane. So you need to remember that certain gases, hydrophobic molecules, small uncharged polar molecules such as water, okay, can diffuse freely from the plasma membrane. It doesn't require any channels or transporter protein. It can diffuse freely from the plasma membrane. But large polar molecules such as glucose or any other large polar molecules, okay, and charge and ions, charge molecule, irrespective of the size, okay, charge molecules and ions cannot diffuse freely from the plasma membrane, but it requires specific transport protein on the plasma membrane. So here we talk about the role of the transport protein. Okay, so transport protein basically plays a role in the transport of these large uncharged polar molecules as well as ions and charged molecules. Okay, from the inside to the outside of the cell as well as from the outside to inside. So when we talk about the transport protein, there also we talk about there are also different categories of transport protein. Okay, so one is active transport and another is passive transport. So when we talk about the active transport, there is a transport of ions against the concentration gradient. Okay. So if your ion molecules are lower inside the cell and higher at the outside. Okay. So let's talk about passive transport first. So in passive transport, basically the flow of ions are done by the concentration gradient. So if the ions are present, higher ions are present in the outside, and the inside there is low amount of ion concentration. Then by these channel proteins, the ion will get diffused laterally from the outside to the inside. Okay, based on the concentration gradient. Okay, it is not a single flow. Okay, if the concentration of ion in the inside is higher, then these channels will form diffuse the ion to the outside. Okay, it is not unilateral in direction. It can be bilateral. Okay. So over here, ion channel is simple. You do not require any energy, okay, because it flows through the concentration gradient. But in the transporter, basically, you need energy, okay, because the flow is from against the concentration gradient, okay, from the lower concentration to the higher concentration. So in that case, you require energy, okay, to transport that molecule. So that is called active transport. Active transport means it requires energy for the transport. Passive transport, it means it doesn't require any energy, okay. And yeah, so these are the difference. So in the active transport, which we when we talk, then there are certain ion transporter proteins present which uses energy. In cell, in case of cellular system, ATP is generally used as an energy source. Okay, but apart from ATP, there are other sources as well. Okay, not it is not fixed that only ATP will be providing the energy. Energy can also be derived from other sources. Okay, other ions which are present higher in the higher concentration inside the cell. Okay, so that will gonna also be generating the energy, the transport. So these are two different transporter proteins, transporter and channel proteins present. Okay, to transport the ions. Okay, so this that constitute the cellular part of the cell. Okay, so this that thing includes the cellular organization, that it is considered the plasma membrane and all the other things and the transporter and all proteins present. Now, when you talk about the metabolism part of the cell, so in metabolism, as we know, glycolysis and glycolysis pathway is an important pathway, and a glycolysis combined with Krebs cycle and cellular ATP generation by the mitochondria. Okay, so this constitute the important pathway by which the ATP is generated. Okay, energy is generated. So from the glucose molecule, basically glucose is converted into pyruvate, and from the pyruvate, acetyl CoA is formed, and this acetyl CoA goes through the Krebs cycle, and from this Krebs cycle, generally the reducing equivalents are generated. So NAD plus is converted into NADPH. Okay, it is reduced. Okay, and it also provides ATP molecule, from it converts the ADP into ATP. Okay, so this NAD plus is converted into NADH, and also FADH2 is converted. Okay, and it provides the energy. So now this NADH and FADH2 get transport to the electron transport chain present in the mitochondria. Okay, so NADH and FADH2 formed during the cellular during the Krebs cycle are donates the electron to the individual complex. For NADH it is complex 1, and for FADH2 it is complex 2. Okay, so for a single NADH molecule will donate 2 electron, and in that process four hydrogen ions will be translocated to the intermembrane space of the mitochondria. Okay, in case of FADH2 they also donate the electron, but in this case the hydrogen ion cannot get translocated in the intermembrane space. So this electron basically it transfers to the various transport chain. Okay, it translocate to the transport protein, and it converts the hydrogen ion and react with the oxygen. Okay, to form a water molecule. Okay, the complex 4. So complex 4 makes the water molecule by using this electron, and it also translocate the two hydrogen ions. Okay, so complex 1, 3, and 4 translocate the hydrogen ions. In complex 1 and 3, 4 hydrogen ions are translocated, and complex 4 is 2 hydrogen ions are translocated. So why I'm talking about hydrogen ions? Basically, this hydrogen ions concentration of these hydrogen ions helps in the uh, formation of the ATP molecule by electromotive force. Okay, so electromotive force is generated by the uh, formation of these hydrogen ions, concentration of the hydrogen ions inside the intermembrane space. Okay, and due to this higher concentration, these hydrogen ions by the ATP synthase enzyme. ATP synthase enzyme will use this electromotive force. Okay, by using the energy from these hydrogen ions, and it will convert these hydrogen ions into the energy by converting the ADP to ATP. Okay, so this constitute the synthesis of the ATP by the ATP synthase. Okay, present in the intermembrane space of the mitochondria. So these electromotive force produced by hydrogen ions are used to move this motor protein. Okay, and by movement of this motor protein, this ATP is converted into ATP. Okay, and this ATP then can be used in the again can be used in the various cellular metabolism pathway and all those things. So uh, by by producing the reducing equivalents such as NADH and FADH2 and donating the hydrogen ions, okay, we generated the ATP. So these are the important process occurring in the mitochondrial electron transport chain. Okay. So next thing is when we now talk about the DNA. Okay. So we'll be now talking about the constitution of the DNA and various other organization. Okay. Yeah. So in the DNA, basically there are four bases present. Okay. Cytosine, guanine, thymine, and adenine. Okay. So there are uh, two chemical components. Okay. One, so cytosine and thymine base. If the cytosine and thymine are generally called pyrimidine. Okay. And guanine and adenine is called purine. So when we talk about purine, they consist of two aromatic chain. Okay, one is a six carbon unit, and another is five carbon unit. Okay, so it contains a six plus five carbon unit aromatic ring, and in guanine basically it contains oxygen. Okay, and NH3 group, and in adenine oxygen is absent, only NH3 is present. Okay, and in pyrimidine, when we talk about the pyrimidines are cytosine and thymine, and the difference are basically the presence of the CH3 group. Okay, so in thymine, CH3 group is present, and CH3 group and the additional oxygen atom. But in cytosine, there is an NH3 group present. Okay, and CH3 group is not present, and it is a single six carbon unit. The purine are 11 carbon units. Okay, 11 carbon aromatic ring. So bases and sugar molecule. So DNA are made up of three component basically. It is called nucleotide. Okay. So if you consider bases plus sugar, it is called nucleoside. Okay. And when we attach the phosphate group, it is called nucleotide. Okay. So this is the thing. And in DNA, basically 2' deoxyribose sugars are formed. Uh, is uh, is present. Okay. 2' deoxyribose. So at the C2 carbon position, C2 dash carbon position, oxygen is absent. Okay. So in RNA basically it is made up of two ribose. Okay, two dash ribose. So ribose. So in over here it is deoxy because the oxygen is absent at the second position. Okay, so this is the deoxyribose sugar which is present, and at this five prime end basically your phosphate group will be attached. Okay, during the constitution. So basically, when we talk about the composition of the nucleotide, so at the first position, C1 carbon position, your base will be getting attached. Okay, so individually the nitrogen atom which are present over here. Okay, so nitrogen atom present in this case, the arrow you can see that nitrogen gets attached to this OH position, C1 OH position, and at the C5 position, your pyrophosphate will be getting attached. Okay, and that is how it constitutes the single nucleotide subunit. Okay, so a single nucleotide subunit is this adenine. For example, this is an example of adenine. So adenine is attached to the C1 position. Okay, and which is attached to the deoxyribose sugar, and its C5 phosphate group is present. Okay, PO4 phosphate group is present, and then this is another moiety. Okay, this is another adenine moiety. So this is another adenine moiety. Okay, this whole component, a nucleotide subunit. So now between the nucleotide subunit there is a phosphodiester bond. Okay, so single unit, uh, the single unit. Okay, uh, single nucleotide unit will be attaching by another nucleotide unit by phosphodiester bond. So this phosphodiester bond is formed between the phosphate present at the C5 position and the oxygen OH group present at the C3 position of the deoxyribose. Okay, so C3 position deoxyribose and C5 position phosphate creates the phosphodiester linkage between the nucleotide. So now this is called dinucleotide. Okay, so attachment of two nucleotide subunits is called dinucleotide. And similarly, when we keep on adding the nucleotide subunit, in which the OH group over here reacts with this phosphate group and with the sugar side chain, it considers the trinucleotide. And similarly, the DNA molecule is formed. Okay, then it considers oligonucleotide. Okay, so oligonucleotide means many nucleotides joined combinedly. Okay, and that it will constitute the nucleotide. So now, when we talk about the genetic information, okay, apart from that, like when we talk about this nucleotide subunit, so this nucleotide subunit is considered the single DNA molecule. Okay, so in DNA molecule itself, there is a pairing between the. So when we talk about DNA, it is a double-stranded DNA molecule. So in the double-stranded DNA molecule, basically, there is a complementary pairing between the purine and the pyrimidine. Okay, so Purine and pyrimidine will a single purine will be forming an hydrogen bond between the pyrimidine molecule. Okay, so pyrimidine hydrogen pyrimidine pairing will not observe, as well as pairing will not observe. Okay, for example, adenine pairs with thymine. Okay, so adenine pairs with thymine and guanine paired with cytosine. So between the guanine and cytosine, there are three hydrogen bonds. Between the adenine and thymine, there is a two hydrogen bond. Okay, okay. So this you need to remember. And according to Chargaff's rule, okay, Chargaff was a scientist, and what he observed was that the amount of adenine to thymine and guanine to cytosine are similar. Okay, so the guanine cytosine ratio and the adenine thymine ratio are similar to each other in a single DNA molecule. Okay, so it is a ratio 1:1. Okay, in the DNA molecule. So this is the information regarding the DNA molecule. So now after the DNA molecule, now this DNA gets transformed into the protein. Okay, so we already know that DNA is not the functional part of the cell. Okay, functions are generally produced by the protein molecule. Okay, but people were confused that initially when didn't know that how this protein is getting made. So people were confused that from where this protein is getting built. Okay, so there is a one example present over here, that in the sickle cell anemia, when we observe, so sickle cell anemia is linked to the S allele of the beta globin gene. Okay, so in the DNA molecule, in the beta globin gene, in the S allele, if there is a single mutation, okay, point mutation in the DNA molecule, so there is in the sickle cell hemoglobin. Basically, if I talk about the general idea, in the sickle cell, there is a single amino acid chain from glutamic acid to valine. Okay, so because of this single amino acid chain, there is a disease condition called sickle cell anemia. So in normal hemoglobin, normal beta globin molecule, there is a glutamic acid, but in the sickle cell disease, there is a valine present. Okay, instead of glutamic acid at the sixth position of the globin molecule, hemoglobin molecule. So when we trace back to the how this mutation is getting done, okay, so scientists found that there is a point mutation in the DNA which changes the codon of a molecule. Okay, so codon, basically, if you know, then codon is a coding part of the DNA molecule. Okay, so for example, three codon. Okay, so three codon, there is a pair of three codon, and this three codon pairs for a single amino acid. Okay, so this will code for a one amino acid. This will be two, and this will be coding for a third amino acid. Okay, and each and every then this code for an amino acid, and then each and every amino acid links with each other to form a complete protein molecule. Okay, but because these amino acids are recognized by this codon, so if you have a mutation in the molecule, okay, mutation in the DNA molecule, so then mutation will be incorporated by the RNA molecule. Okay, and RNA will be converted into protein. So if you have any mutation in this DNA molecule, so that mutation will change the codon. Okay, the codon reading frame of this protein molecule. So if you have a single point mutation as well, for example, molecule such as this is just an example. Okay, so ATG, for example, codes for a one amino acid. Okay, and for example, ACG codes for another amino acid. Okay, so in place of T, you have a C, then you have altogether a different protein molecule. Okay, so in this case, what we are observing, glutamic acid instead of valine. So this is due to that this is done. Okay, due to this change, basically it changes the structure, it changes the amino acid of the protein due to single amino acid change. Okay, and that is how it is called. It is a central dogma of a molecule. Central dogma of a cellular system. Uh, DNA codes for a RNA, and RNA codes for protein molecule. Okay, that is how the information is getting translocated. So during the transcription process, now we will talking about transcription. So transcription is the process from which the RNA is converted into DNA is converted into RNA. Okay, and in transcription, basically, when we talk, so in transcription there are various factors which promotes the transcription. Okay, which regulates the transcription process. One is like chromatin environment. So how chromatin is associated, whether it is loosely packed or whether it is highly accessible region. Okay. So this blue color region you can see that is called nucleosome. Okay. So nucleosome is a protein which wraps this chromatin molecule. Okay, which compacts the DNA molecule basically. Okay, so if the chromatin molecule is devoid of nucleosome, then the transcription factor can act on that nucleo or that chromatin molecule. Okay, so you need to unwrap this chromatin with this nucleosome from this nucleosome, and then the transcription factor will work and the RNA will be produced. So transcription process is regulated by this chromatin environment as well as nucleosome occupancy as well as this histone modification. Okay, so histone, there is certain modification on the histone. So, for example, methylation and acetylation are generally observed. So, if you have a histone modification, methylation on the DNA, so that promotes that the gene will not get transcribed. Okay, so methylation represses the transcription of the gene, and if you have unmethylated region, then that gene can be transcribed. So inversely, if you have acetylation, then in the acetylation, your gene are active, and if you don't have the acetylation, then that gene will not be active. Okay. So histone modification also plays a role in the transcription process, and also the cis regulatory element. The cis regulatory elements with the enhancers. Okay, enhancers sequence present in the DNA region, which enhances the transcription of this transcription factor. Okay, by the transcription factors. Enhancer is that region. So if the enhancer are present, then that gene will be active in that region. So when you talk about eukaryotic gene regulation, the transcription initiation, you have certain factors, okay, various factors present during the initiation process. There is a transcription factor, various factors, 2F, 2E, 2H, okay, and various other activators and mediators. So activators binds to this enhancer element, okay, and mediators are the protein which bridges the activator with the transcription factor. Okay, so as I told, key enhancer will enhance the transcription of the protein molecule, of the RNA molecule, basically transcription of the RNA molecule. Okay, and this by the use of this ATP, it will transcribe the mRNA molecule of the protein. Then you have translation process in which the RNA molecule, the processed RNA molecule, which contains the G capping as well as the 5' capping, okay, as well as the poly A tail. It is called the processed mRNA molecule. So this mRNA molecule is translated into the protein molecule by the help of ribosomal subunits. Okay, so there are basically two ribosomes. Okay, RNA RNA molecule. One is the two RNA molecule, ribosome rRNA as well as tRNA. Okay, so rRNA is ribosomal RNA, and that helps in the binding. It binds to the mRNA. It helps for the recruitment of this tRNA. Okay, so tRNA basically uh, is a transfer RNA in which the amino acids are attached to the it is 5' end. Okay, amino acids are attached, and by recognition of the specific codon on the mRNA molecule by the ribosome, and by that specific tRNA are placed on that codon region. Okay, and amino acids are synthesized by one another through polypeptide through peptide bond formation. Okay, and proteins are formed by that. So when we talk about translation process, there is a codon will get attached first, okay, at the P site, okay, and at the P site it will get attached, and another codon will get accepted at the A site, and the amino acid which are present at the P site, it will form the phosphodiester. It will form the peptide bond between the amino acid present in the A site amino acid, okay, and then it will move further in the elongation step. It will move further, and this A site tRNA will move to the P site, and then further amino acid will be coming, and the whole further tRNA will be coming, and whole protein molecule will be formed. Okay, the chain of amino acid will be formed. Okay, and after a certain condition in the termination stage, it will get terminated. Okay, and the amino acid will be get released, and then this amino acid will be folded into its component, and will we get the intact protein molecule. Okay, so these are the things regarding the transcription translation process. So till now, you have any doubt? Uh, Till now, if you have any doubt, then you can ask. Okay, so after that, we'll be solving an assignment question in the next 40 minutes. Okay, I mean, a 40-minute class is left, so in that, we'll be solving some assignment questions. Yeah, so any doubt, anyone?
Sir, tomorrow the timings of the class are clashing.
What is the timing? What is the timing of the class?
It's 4 to 6 and 3 to 5.
Yeah, so
Two classes.
basically, you can uh, you can attend any of the classes, okay, any of the tutorial classes. So if you've attended this class, then it's not required that you should also attend that class as well, because the general idea of the tutorial session is basically uh, to clear your doubts in that week. Okay, so in the week one session, what you've gone through, so that doubts will be get cleared for your, and the assignment questions will be solving assignment questions which you have already attempted. Okay,
Okay, so
so
from the 4 classes only we have to attend one class.
yeah. You can attend only one class from the any of the tutorial session you can attend. So the content will be discussed is the same. Okay. So apart from that, like if anyone is so in actually this is week one. Okay. So you can you can
And sir, last date
eh,
of the assignment is 7th Feb.
which one? Second assignment.
First assignment also it is given 7th Feb only.
Okay. So first assignment you haven't submitted yet. I think the date was due now for first assignment. For
Yeah, it
second
was 7th
assignment,
Feb only.
7 Feb only.
Yeah.
Okay. So you haven't solved that question. If you have solved the questions or not for assignment one, you haven't done.
Yeah, good evening, sir.
Yeah,
Sir, actually, the first assignment submission date is Feb 7, sir.
7 February.
Yes, sir.
Okay.
Okay, sir. Sir, and I have a doubt like in this week only the tutorial session will be there, or else every weekend there will be a tutorial session, sir.
No, every week the tutorial session will be there. So basically, the content which you will be accessing in that week. Okay.
Mm.
So at the end, we'll be having tutorial session, and if you have any doubt, then we can solve. And otherwise, in the tutorial session, we will revise. Okay, the week content.
So, sir, and yet I didn't enroll myself in the course. Like probably I will enroll on Monday, sir. But I would like to pursue one more course. Is it possible to I can pursue both the courses, sir?
Yeah, you can attempt any of the. I mean, you can enroll to any of the courses. There is no limit to the enrollment. Depending that your basically your tutorials session which we conduct. So so basically your course content are already uploaded on the website, but the tutorial session it should not clash. Okay, so if you're enrolling in two courses and you want to attend the tutorial session of this both the courses, if the time is clashing, then it will be difficult for you. Okay, so depending on that, you
Oh,
arrange your
thank you,
course.
sir. And tomorrow also that is tutorial session is there, but the same topic will be discussed, right?
Yeah, same topic, but what I'll be suggesting to all of you is that you attend that tutorial session. If the time is clashing, then you can attend any one of that, and depending on your depending how you access that, if you learn something new, then it's for your good only. But if it is the same thing, if it is described in the class, okay, in the tutorial session as I discussed, then I don't think there is a point in attending that session from the next week. So this week you can try, okay, as a demo,
Sure,
you
sir.
can attend.
Thanks for the insight, sir. Sir,
Yeah.
and I have only one more doubt, sir. Only weekend we have tutorial session, not in the weekdays for sure.
Yeah, only weekend. For this course is basically on the weekend only. For this course, Saturday and Sunday.
Okay, sir. Thank you, sir. Thanks for your insight and solving all my queries. Thank you, sir.
Yeah. Yeah. So let's solve the assignment questions. Okay. If you haven't solved, then because in the next class we'll be discussing over the assignment two questions. So I think in this tutorial session, we will solve the questions for the assignment one, and in the next tutorial session we will be solving the second assignment question. Okay. So if you haven't attempted, then it's fine. Okay, but you can just go through it. It is simple or simple. But what I'll be suggesting to you, all of you, is that at the start of the week, okay, you go through the lectures first, okay, in that specific week, and you try to solve the assignment question before the tutorial session. Okay, so every Saturday there will be tutorial session of ours, okay, of mine, that is from 3 to 5. So before that, if you solve the assignment question, then it will be better for you in two ways. Okay, because first you will be solving the assignment question, so you will be done with that. And another thing, after solving, we can also discuss that assignment question in our tutorial session. Okay, so if I discuss over here and then you will solve, then it is of no use. Okay, so you need to solve it on your own first. After that only, it will be good for you to for discussion as well.
Yes, sir. We got it, sir.
Yeah. So in this session, we will solve the assignment one question, but in the next session, you try to attempt the assignment question before the tutorial session. Okay, so
Okay, thank you, sir.
yeah, yeah. So
So
let's
thank you, sir.
discuss.
Thank you so much, sir.
Yeah, so the class is not over. We will be solving the assignment question for there is for half an hour.
So, is it a model assignment question or is the question which going to be in our assessment for the week, sir?
This is for week one assignment question. So this is a week one assignment question, and so you can because in the next week we will be solving the second assignment questions. Okay, so the aim of this tutorial is to basically solve the assignment question. Okay, so if we skip this assignment question, then it will be difficult. Okay, in the next class, like we have to arrange that this assignment question. So in my point of view, like if we discuss this assignment one question over here, then it will be good in this session only. Okay. Yeah, so there is 10 questions. Okay, it will not take much time, and uh, first, what we'll be doing is that there is okay, yeah. So this is question number one. Gram-positive bacteria are stained purple with crystal violet because there are four options. So you need to select one option. Okay, so if you have not solved, then you can solve your only. Okay, uh, I mean, by taking paper and pen, you can just write A, B, C, D, and after that you can evaluate on your own. Okay, so I will not disclose the answer before attempting. So after that we can disclose. Okay, answer.
So whether we should try to give answer now, I mean in the class now.
Yeah, you can verbally like tell ki what is the correct answer, and you can also solve in your book. Okay, I mean in one page, and depending on that you can evaluate yourself. And during the assignment submission, you can directly then click okay over there during the assignment submission. But if you want to evaluate currently, then you can just attempt it in your book. Okay, in your page, one single page, and then you can at last you can evaluate yourself. So what we'll be doing is you can give the answer out of this four option, and then we can discuss the which is the correct answer. So what will be the answer for this question? Gram-positive bacteria are stained purple with crystal violet because they have a thick peptidoglycan layer and no outer membrane. They have a thin peptidoglycan layer. They have a thick peptidoglycan layer and thin outer membrane, and they completely lack the peptidoglycan layer but have the outer membrane. So what will be the
Option
correct
C.
option? Option C. Option C.
Yes, sir.
So what others are suggesting? I want at least 5 to 6 answers. Okay, you are total 20, 21 student. So at least 5 to 10 answers I need collectively. And that way only, that is the basically the idea of solving the assignment questions. Okay, it's not like I will show you the answers and show the solutions. You need to remember the content from the slide or from the that week, and then you have to solve that question. Okay, so that is the rule. So please give at least 5 to 5 answers. Others can answer. What will be the correct answer over here?
Option A.
Sir, can I change my answer?
Sorry.
Can I change my answer?
Yeah, go ahead. You are always uh, welcome. So these we already
It's
discussed.
option A. Option A, because I didn't notice that option A. I didn't gone through. It's option A.
Option A. Others. I got two answers. Others. I need answers from at least three student. Others three student.
Sorry.
Option A.
Sir, option A.
Okay. Anyone? Others? Okay. So option A is the correct answer. Okay. They have thick peptidoglycan layer and no outer membrane. So if I talk now regarding the yeah. So if I talk for gram-negative bacteria. Okay. So why gram-negative bacteria stained with pink? So what will be the answer over here?
Because they have thin peptidoglycan layer.
Thin peptidoglycan layer. And what about the membrane?
Outer membrane is present.
Outer membrane is there. Okay. So that is
Yes,
the
sir.
regarding the gram-negative bacteria. Okay. So this we discuss in the slides. The crystal violet during the crystal violet staining because peptidoglycan is very low in the gram-negative. It will be staining by the pink in the by safranin. Okay, due to safranin, secondary staining. And in crystal violet, it will be staining very high. Okay, in the gram-positive bacteria. So now second question, which of the following statement doesn't hold true for the nucleolus? So anyone know about what is nucleolus? What is
Which is
nucleolus?
for RNA synthesis at present inside the nucleus.
Yeah, so it is a site for ribosomal RNA synthesis. Okay,
It is responsible for rRNA synthesis.
yeah, that is your ribosomal RNA. So out of these four options, which is incorrect? So you have to pick up the incorrect option. So out of these three are correct, one is incorrect. So please answer.
Option C.
Option C. It is bound by a thin membrane. Okay. Others?
Option D.
Option D. Site for ribosomal assembly. Okay. One, two,
I
one.
was confusing with the assembly of the ribosome because they have two subunits. Uh, they
Hmm.
got confused.
So what other students are suggesting? What should be the correct answer over here?
Option B, sir.
Option B. B or D?
B.
Okay. Option B is you are telling it is an incorrect. Okay. Others. So we have got three different options by three students. What are others suggesting? The majority. So you have to answer now. Okay. We need to derive majority over here in the single option. Okay. Please participate. Okay. Otherwise, it will not be interesting. So other student, please. You can. No one is judging you over here. Okay. And no one knows who are you. Okay. You are behind the screen. So please engage yourself in the class. It will be interesting for you as well as it will also beneficial for me as well. Okay. Yeah. Go ahead. Please answer someone.
C, C.
Option C. It is bound by a thin membrane. Okay. So please don't use internet. Huh? So you need to be no cheating over here. Yeah. So other two, three students at least I won't answer. Option C we got from the two student. Option B and D.
Sorry, sir.
We need answer from more other two student at least. Yeah, go ahead. What could be the correct answer?
Option C.
Option C. Okay. So option C is the correct option. Okay. It is bound by thin membrane. So basically in the nucleolus, when we talk about nucleolus, okay. So nucleolus is a site for the ribosomal RNA synthesis. Okay. So various subunits are first it is transcribed. Various rRNA are transcribed from the DNA molecule. Okay. From the ribosomal DNA molecule, it will transcribe into ribosomal RNA. And then after transcribing that assembly processing that transcription processing and assembly occurs in the nucleolus. Okay, so ribosomal RNA transcription, rRNA processing and the subunit assembly occurs in the nucleolus of the cell. Okay, and so basically in the nucleolus is formed. Okay, it is assembled at the end of the mitosis phase. Okay, and it recruits this certain transcription and processing machinery which is helpful in generating the ribosomal subunits. Okay, so at the end of the mitosis this nucleoli is formed and it contains ribosomal DNA molecule from which the ribosomal RNA will be formed and other by having the transcription factors and other post-translational post-transcriptional mechanism inside the nucleoli. The complete processed ribosomal RNA is formed. Okay, and after that, the assembly will also occur in the ribosome in the nucleoli. So then these nucleoli, what it will then this nucleoli helps uh, in the translation process in the protein during the protein synthesis. Okay, so this is this. So it is a dense body present inside the nucleus. It is correct. It is bound by thin membrane. It is a site for rRNA synthesis. It is correct, and it is a site for ribosomal assembly. So these three options are correct. It is not bound by thin membrane. Okay. So when we talk about the structure of the nucleolus, then the nucleolus lacks a surrounding membrane. It is comprised of three main components. Okay. So it doesn't have the nucleolus doesn't have a membrane around it. Okay. But it has three components such as fibrillar center, dense fibrillar component, and granular component. Okay. So this is slightly in detail, but you don't need to worry about what is occurring in which component, but this is just a general information key. It doesn't have any membrane. Okay, it has three different components and these three components actually are involved in the different stage of ribosome production. Okay, so we will not go into detail key which components are involved in which stage. Okay, and in the nucleolus basically the DNA, RNA and other proteins which help in the transcription process and translation are present. Okay, and it is a site for ribosomal protein synthesis. Okay, and synthesis and assembly, all the three, transcription, synthesis, and assembly, all the three processes occurs in the nucleosome, nucleoli. Okay, and regulation is basically dependent on the cellular demands of the protein synthesis. Okay, so if the cellular demands of the protein is higher, then there is a higher activity of these nucleolus. Okay, because there will be a requirement of ribosomal RNA over there. Okay, during if the protein synthesis demand is higher, then rRNA will be required in higher amount. So due to that, the that is how it is regulated. Okay, so in the resting condition, basically the nucleolus role will be not be very high. Okay, and in the certain disease conditions such as cancer and all those things, okay, there is a in the genetic some genetic disorder there is an aberrant function of this nucleolus. Okay, so in cancer generally it is a excessive proliferation of cell, and due to excessive proliferation the protein synthesis will be also be higher, and at that stage the nucleoli will be having there will be function there is a aberrant function of this nucleolus activity. Okay, during that condition. So this is the general idea of the basic idea of the nucleolus. Okay. So now let's move the third question. It is simple question. Okay, so solve uh, solve yourself. Okay, what will be the correct option over here?
Sir A.
Okay,
Option A.
option A. Others?
Option A, sir.
Sir, A.
Okay. A. Yeah.
B, sir. Option B.
Option B.
Yes.
Why option B? Any reason? Any reason why option B? The only way to cross the cell membrane is via the transporter protein. Okay, let's consider option B as well. Others?
Sir, option A.
Okay. Yeah, so correct option is option A. Okay, so it allows the free diffusion of gases, hydrophobic and small uncharged polar molecules such as water, and also contain transporters to selectively transport large polar molecules such as glucose. So this is the option, correct option. Option B is the only way to cross the cell membrane via transporter protein. No, basically your gases and hydrophobic as well as small uncharged polar molecules doesn't require the transporter protein. Okay, it can freely diffuse from the plasma membrane to and fro. Okay, so it is a way for the transport. Transporter proteins are required for certain ions as well as large polar molecules, but it is not the only way. Okay, so that this option is incorrect. It completely restrict the movement of large polar uncharged molecule. No, it can allow the movement of the molecules, large polar molecules, by using the transporter protein. Okay, so option A is the correct option. So during solving the MCQs as well, you need to uh, basically look into the you need to know the basics very clearly. Okay, during terminologies, and if you have slight doubt, then generally in the MCQ what it does is that if you have a slight doubt, then also you can choose the incorrect option. Okay, so your theory should be correct, concept should be clear over there. So this I already explained to you, okay, in the previous slides. So these gases and hydrophobic molecules are generally passes through the diffuses freely, but transport proteins are used for the ions as well as large uncharged molecules. Okay, so this is the basic idea. Okay, so in ion canals and ion transporters, if your ion transporter are basically used when you transport the protein, transport the ions against the concentration gradient, and ion channels allows the transport through the concentration gradient. Okay, down the concentration gradient. Okay, question number 4. Pick the correct statement. So note
Option
your
A.
option
Sir,
A.
option A. Guanine is a purine base. Adenine and guanine is a purine base. That's a correct answer.
Okay,
Yes, sir. Option A.
okay, option A. So option A is correct. So this we talked, okay. Pyrimidine constitutes cytosine and thymine. Purines are guanine and cytoadenine. Okay, and yeah, so what it says, guanine is a purine base. Yes, guanine is a purine base.
Can you
Okay,
please explain the carbon position of this ribose sugar?
ribose sugar carbon positioning.
Yes, sir.
So what I need to explain key where base or phosphodiester bond phosphate is attached.
No, with the help of the carbon position only, we are finding the polarity of the DNA molecule. I still having the confusion about that. Could you please clear it?
Yeah, so yeah, I got your question. Yeah, so basically, it is a 2' deoxyribose. Okay, so in general, the sugar moieties are two ribose sugar. Okay, ribose means it contains an oxygen. Okay, oxygen present. Okay, OH group present in the second carbon position. But in the case of DNA, it contains a deoxyribose. Okay, at the second position. So that in that case, oxygen is removed. So this is the positioning. This is carbon number 1, 2, 3, 4, and 5. Uh, 5 over here. Okay, and over here there is a O bond. Okay, oxygen present at the fourth position, 4 prime position. So at the first position, base attached at the first position OH group, and at the fifth position phosphate group is attached. Okay, so polarity is generally induced in the 5 prime to 3 prime direction. Okay, so when we talk about DNA, it had a 5 prime phosphate and a 3 prime OH. So when we talk about this deoxyribose, so you can clearly see key at the 5 prime position when it makes up the nucleotide, this phosphate group is attached at the OH position. Okay, so this constitutes now the 5 prime position of the phosphate. Okay, at the 3 prime position, OH is present. Okay, and this OH now will make the phosphodiester bond with the incoming nucleotide. Okay, so that I already had shown over here. Uh, okay, so hmm. So this is the adenine residue. This is the deoxyribose. Okay, 2' deoxyribose, 2 prime deoxyribose, and this is the phosphate group attached to the fifth position. Now the incoming nucleotide will make a bond. So it already incoming nucleotide means it contains the phosphate group at the 5 prime position. And over here we have OH group at the 3 prime position. Okay, third carbon position. So this OH group will act as an electrophilic agent. Okay, it will donate the electrons and to make the phosphate group. Okay, phosphate it will make the phosphodiester bond by donating the electron. Okay, so at this the basically the phosphodiester linkage is formed by the C5 prime position of the deoxyribose sugar and the 3 prime position of the already present deoxyribose sugar. Okay, so 5 prime position phosphate of the incoming nucleotide and the 3 prime position of the already present nucleotide, and this will make the
So that
phosphodiester.
when the third position is doesn't contain the phosphate group, that is a 3 dash. And when the fifth position is occupied with the phosphate group, that is a 5 dash. So the opposite will be the quite opposite for if it is 5 dash, the opposite will be the 3 dash, and the 3 dash, the opposite will be 5 dash.
Yeah,
Is it
yeah,
correct? Sir?
yeah. So over here, basically, if the nucleotide is free, okay, if it is not linking with the phosphate group, then it is called 3' OH. Okay, but if it is getting linked by the 5 prime position, then it is not a 3 prime OH group. Okay, it considers the phosphate group now.
Yes, sir.
If
Yes,
it,
sir. I got it. Thank you, sir.
yeah. So if it is free only, then we can say that it is a 3 prime OH. If it is added, then it is not a 3' OH group. Yeah. Okay, so where were we? Over here. Yeah. Yeah, so this guanine is a purine base. This is a simple question. Okay, so now regarding the Shargov's rule. So this question, attempt it.
Sir, option C.
Option C. Okay. Any other?
So option C.
Okay. Others? I need three, four more answers.
Sir, option C.
Okay. Option C.
Sir, option C.
Okay. Yeah, so option C is correct. The ratio of A to T and G to C are close to unity in the DNA. Okay. So this is the correct option. And you already know about the Shargov's rule. Okay. So in the Shargov's rule, basically base pairing rule is adenine pairs with thymine and guanine pairs with cytosine. Okay. And the base composition of A to T and G to C is 1:1 ratio. Okay. According to Shargov's, this should be the ratio of A to T and G to C. Okay. So if we, yeah. So next question is this: which of the following organisms belongs to the prokaryotes? So you need to answer.
Archaea bacteria.
Archaea.
Archibacteria.
Okay. Others?
Yes, sir. C is the option. Archibald.
C. Okay. Yeah, so Archaea is the answer. So we talked about this as well, prokaryotes and eukaryotes. In prokaryotes, generally it is unicellular. Okay. So in that, eubacteria and archaebacteria are the prokaryotes, and in eukaryotes, animal, plants, yeast, fungi, and protist. Okay, so these are the eukaryotes, which contains nuclear membrane. Okay, and it is a multicellular as well as unicellular as well. Okay, and it contains various other organelles like mitochondria and centrosome. Okay, and it is a linear DNA. So archaea is a kind of prokaryote organism. Yeah, so question number 7 regarding the carbon atom. What is the correct option?
Sir, option B. Carbon can form 4 covalent bonds.
Yes. Even in
Mhm.
amino acid. Yeah, option B, sir.
So what about third option?
So there is no upper limit to the size.
Okay.
Actually, I'm not aware much about that, but as it's a very basic thing, I go with option B. But I'm
Mm-hmm.
not aware of option C.
Yeah. So others, what others think? Yeah. Anyone? Please answer. So I got two options. Option two, option B. I got two answers, two responses. Others, or is it
Sir, option A.
option E? Carbon forms unstable carbon-carbon bond. Okay. Anyone? Others? You need to respond. Okay. So option B is the correct. Carbon can form four covalent bonds. Okay. So B is the correct answer. because as I told ki it has a sp2 hybridization. Okay, so there are four unpaired electron present at the outer shell of the carbon. So that can pair with the four. It can make the four bonds. Okay, four covalent bonds with the neighboring elements. So option A, when we talk carbon form unstable carbon-carbon bond. No, the carbon-carbon between the carbon-carbon bond, it is a covalent bond. Okay, and covalent bond is not a unstable bond. It is a stable bond. And regarding the third, there is an obvious upper limit to the size of the rings formed by carbon atoms. No, carbon can form a multiple number of side chains. Okay, sides of the rings or chains. Okay, so there is no upper limit for the formation of this carbon molecule. So this is general basic idea कि carbon is a tetravalent molecule, that it can form four covalent bond, and carbon can have a unique property to have form long chain and branch chains. Okay, so that property is called catenation. Okay, so catenation is basically the bonding of atoms of the same element into a chain. So if there is a single carbon chain, so that process is called catenation. Okay, so this chain can be a ring shape, or that is called open chain, as well as that can also be closed. Okay, that is called cyclic chain. Okay, so open or closed chain. Okay, they can be both kind of chains. So catenation basically can form with the carbon, which forms a covalent bond. Okay, or any other molecule which forms a covalent bond, and it makes a single chain molecule. Okay, so that is the process known as catenation process. Okay, so if you go through, I mean, if you come around this term, the carbon can make a catenation. Okay, so you need to know that what is catenation. Okay, so that can also be helpful to answer the question. Okay, so question number 8, last two question. Which of the following allows the ion to diffuse down the concentration gradient? Ion
Sir,
transport.
ion transporters.
Okay, any other? Oh,
Sir, down means against the concentration gradient.
no, that you need to first you answer it, and then we will discuss the down means what. Yeah, so basically downs means from the concentration gradient, not against.
Sir, it is option B, ion channels.
Okay. So you need to understand, okay, the terminologies.
I think is it ion channel is against the concentration gradient, right?
Okay. We'll see. Let other answer. Yeah, other response. What I can go through is that only 4 to 5 students are responding in the class of 20. So other 15 students are, I don't think how, because if you don't engage, then it will not be, you will find it boring. Okay. So please respond. You can choose any of the options. Okay. As I told, no one is judging you, but you need to be engaging in the class. Yeah. Response. Any other response? Which the following allows the ions to diffuse down the concentration gradient. So first thing is that what diffuse down the concentration gradients means. Okay. So diffuse down means uh, through the concentration gradient. Okay. So one is the term called diffuse down, and another is diffuse against. Okay, so diffuse down means through the concentration gradient from higher concentration to lower, and diffuse against the concentration gradient means the lower concentration to higher concentration. So which is the correct option? Basically, ion transporter, what it does? Option B. Ion channels.
Sir, B.
Yeah. So ion transporter wave, what it does? Ion transporter diffuses against the concentration gradient. Okay, from lower concentration to higher concentration. So please remember that if you are in any physiological terms, okay, if you are transferring some material from the lower concentration, from its lower concentration to higher concentration, you will always require energy. Okay, energy is always required from transferring the lower to the higher concentration. But during the transport from higher to the lower, it does not require energy because it is already higher. So it will passively diffuse through the channels. Okay, so the process which require energy over the transporter are used, but the process which doesn't require energy, okay, for which directly transport the ion, that is called channels. Okay, so you need to remember in this case, channel means direct passage, and transporter means it requires a certain energy. So if energy is required, then it might be transferring the ions from the lower to the higher, and channels mean direct passage means it will be transferring from the higher to lower. So in that sense, you can remember so that you cannot be confused. Okay, in that term, what is transporter, what can transporter do, and what is channel can do. So yeah, so ion channel is the correct option. Last is the bond between the nitrogen base and the sugar group is nucleotide. In a nucleotide is termed as
Phosphodiester bond.
okay,
B.
phosphodiester.
Phosphodiester.
Okay,
Option
so
A, sir.
option A, phosphodiester linkage.
So it is B, sir. N-glycosidic bond.
Yeah, what happened? Phosphodiester linkage. It is the nitrogen base and the sugar group. Phosphodiester bond is formed between
Sir,
which
I'm
molecules?
sorry to. Hello, sir.
Yeah,
Sir, I'm sorry to ask. Like, is this
sorry, you are not audible. Hello, hello.
Sir, if it's sugar and nitrogen nucleoside, then it is N-glycosidic bond.
Yeah, the phosphodiester bond will be formed between which residues? That I'm questioning.
Phosphodiester bond is formed between two nucleotides. So
Between
it
the
will be N-glycosidic bond because it's between
no,
sugar
it will
and
form between the two nucleotides.
nucleotide.
That I got, but specific, if you want to be specific, then between the
I didn't
phosphate
got.
and between the phosphate and the sugar group. Okay, if the bond is
Yeah, that
between the
N-glycosidic bond.
yeah, so if the bond is between the phosphate group and the sugar group, that is called phosphodiester linkage. Okay, the phosphate of the incoming nucleotide and the deoxyribose nucleotide or deoxyribose. Okay, start prime position. That is called phosphodiester linkage. So over here, it is a N-glycosidic bond. Okay, between at the C1 position between between the adenine and between the nucleotide and the deoxyribose sugar. Okay, urine and the deoxyribose sugar. That is called N-glycosidic bond. So you need to remember this thing. Okay. So last question: in an RNA molecule, urasis replaces which of the following base in the DNA?
Option C. Thymine.
Okay, so thymine
I
is the correct answer. So uracil is basically replaced with thymine, and other adenine, cytosine, and guanine remains as it is. Okay, so these are the assignment questions. So yeah, so I think you haven't solved that assignment question. So you have already noted, okay, in your in your page. So in that way you can evaluate yourself. Okay, and yeah, so that is all from this. So if you have any doubt other apart from this, okay, if you have any doubt, then you can ask regarding this lecture.
So you were saying something about the cancerous cell in nucleolus. I didn't got that point.
Yeah, so nucleolus basically in the cancer cell, sorry, in the cancer cell basically what happens is that the protein synthesis is required higher. So cancer means uncontrolled proliferation. Okay, so if the
Yes, sir.
cell is getting uncontrolled proliferated, so in that case, synthesis of protein should be requiring is higher in amount. Correct. If the cell
Yes,
number
sir.
is high, then protein synthesis will be higher in amount to regulate its metabolism and its function. So because of that, the protein synthesis basically rRNA helps with the protein synthesis. Okay, ribosomal RNA. So you need to have the active ribosomal RNA. So transcription as well as processing and assembly should be there, proper assembly and all for the ribosomal RNA. And ribosomal RNA assembly and all those things are present in the nucleolus. So the nucleolus should be very active in that case in the cancer cell, so that it will make proper rRNA molecule, and after that only the protein synthesis will be done.
Okay, sir. Thank you, sir.
So if you have any other question, you can ask. Otherwise, we can conclude this session. Okay. So thank you all of you, and we will be meeting next Saturday. Okay. So apart from that, please go through these week lectures. Okay, it is already available online. So go through these week lectures and attempt the questions. Okay, assignment question before the next week class. Okay, so after that we will be having a good idea in solving the assignment question. Yeah. Thank you, all of you. Thank you.
Thank you, sir.
Yes.
Thank you, sir.
Yeah. Yeah.